After studying this module, you shall be able to learn the basic PCR technique, understand the principle underlying the PCR, understand the applications of PCR, comprehend the modifications of basic PCR technique. The basic phase of discoveries and understanding of modern biology, especially in the areas of genetic engineering, can be attributed to the formation of high skill technologies. Polymerase chain reaction, called as PCR, is one of the most influential molecular biology technique that is used to amplify genes or genetic transcripts. Using PCR technique, a single or few copies of a particular DNA sequence can be amplified into thousands of copies with the same DNA sequence in a matter of several hours only. The technique is based upon thermal cycling, consisting of multiple repetitive cycles of lowering and raising the temperature of the reactions for DNA denaturation, followed with enzymatic DNA replication. Initiation of PCR, it requires short stretches of nucleotides or DNA fragments called primers containing DNA, RNA sequences which should be opposite to the target region. A DNA polymerase, which is an enzyme after which the technique is named, this enables selective and repetitive cycles of amplification of DNA which is also added. As the PCR further progresses, the DNA synthesized is on its own now, which is now used as a template strand for replication, thereby leading to a chain of reaction that results into exponentially intensification of DNA template. A large number of genetic manipulations can be made by exclusively modifying PCR. The most vital requirement for PCR is a heat resistant DNA polymerase, for example, the Taq polymerase. This enzyme was isolated from the bacterium Thermus aquaticus. As the increase in the temperature causes an irreversible loss in the enzymatic activity for most of the enzymes, the Taq polymerase retains it because it is thermostable. A mixture of nucleotides that are the building blocks of the DNA, single stranded DNA which acts as the template, and the oligonucleotide DNA primers, when enzymatically react with the DNA polymerase, then it leads to the synthesis of a new DNA strand. Most of these PCR methods use thermal cycling, which involves alternate increase and decrease in the temperature of the PCR sample through a planned chain of temperature steps. Thermal cycling steps are essential to denature and physically split the bond of the two strands of the DNA double helix at a predefined high temperature that causes the division of the two strands. This process is called DNA denaturation or DNA melting. Following the melting, the temperature is decreased and then each strand acts as a template strand for the synthesis of a new DNA via the DNA polymerase enzyme that selectively amplifies the template DNA strand. PCR is a highly selective technique leading to multiplication of only a specific DNA sequence, which is achieved as a result of the use of primers that are complementary to the target DNA region used for amplification. It was developed by Carey Mulliez in 1984. Mulliez, a chemist at Cetus Corporation, used his idea to use a couple of primers to bracket a DNA sequence and to replicate it by using DNA polymerase. So, he finally developed a method that allows a small strand of DNA to be copied into ample number of times. Soon after, the importance and the applications of this technique were realized in molecular biology, and in 1993, Mulliez was honored by the Nobel Prize in Chemistry, a great achievement. In modern biology labs, PCR is a highly common and indispensable technique attributed to a variety of application it offers. Some of the most common applications include DNA cloning, DNA sequencing, DNA based phylogenetic analysis, etc. In addition, PCR can also be used for the diagnosis and the cure of inherent disease, the identification and authentication of genetic fingerprints, paternity testing, and also for the analysis of infectious disease. Further modifications of PCR such as the RT-PCR, that is the real-time PCR, offers more specific applications and have resulted in making PCR an indispensable element of any laboratory. The thermal cycler. A thermal cycler is a laboratory equipment that is used to multiply the target DNA segments by means of PCR technique. It is commonly called as the PCR machine or the DNA amplifier. A thermal cycler consists of a heating block with slots for holding tubes which contain the desired reaction combination. The block is thermocoupled and its temperature can be raised or reduced by using pre-programmed steps. A thermal cycler can contain additional heating block which allows using more number of reactions to be carried at once. In addition, in several instruments, a gradient of temperature can be formed in different regions of the block. This particular function is very useful as it helps in optimization of the PCR while checking different sets of primers. Although rarely, but these instruments can be used to carry out temperature sensitive reactions such as restriction digestion, etc. Thermal cycler instruments are relatively inexpensive and are also portable. So, they are convenient and require very less space, and also they offer a wide array of applications. Now, students, let us study the principle of polymerase chain reaction. As mentioned before, PCR is used to identify a definite region of a DNA strand called the target DNA. Using several advanced PCR techniques, amplification of DNA fragments up to 45 kb in size can be achieved. A basic PCR setup consists of the following components: template DNA. This template DNA contains the target DNA region to be amplified, primers, two primers which are alternate to the 3 end of the sense and the antisense base pair of the DNA target, heat stable DNA polymerase. Any heat resistant DNA polymerase having an optimum temperature at just about 70 degrees Celsius, just as the Taq polymerase. Deoxynucleoside triphosphatase. These are commonly called as the DNTPs that act as the basic component for the fusion of a new DNA strand by the DNA polymerases. These include the DATP, the DGTP, the DCTP and the DTTP. Buffers. To maintain the suitable pH throughout the amplification reaction that in turn is required for the optimal activity of the DNA polymerase. This buffer solution is also used to provide a chemical environment that is required for fidelity of the DNA polymerase. And the last, we also require ions such as these divalent cations, namely magnesium, or manganese ions such as Mg2+, which is used because higher Mn2+ concentration increases the chances of error in the course of DNA synthesis, but Mn2+ can be utilized for PCR mediated DNA mutagenesis. Monovalent cations also play a significant role and are provided by potassium ions. Polymerase chain reaction is generally run in a mixture volume of 5 to 200 microliters in small reaction tubes which are made of polypropylene and are thin walled, thereby allowing rapid exchange of heat in a thermal cycler. The required temperature is achieved by alternate raising and reducing the temperature of the reaction tubes during each step. Majority of the recent thermal cyclers are based on the principle of the Petlier effect that allows both the increase and the decrease in the temperature of the silver block that holds the PCR tubes by simply reversing the path of flow of the electric current. Positive thermal conductivity accounting to quick thermal equilibration can be easily achieved in thin walled reaction tubes. In older models of the thermal cyclers, a coat of oil used to be added to avoid condensation at the lid of the reaction tubes because of the lack of heated block for the lids. However, most of the current instruments use heated lids so that it does not allow the condensation to occur at the top of the reaction tube. Procedure. The PCR generally requires a succession of 20 to 40 repeated temperature variations called as the cycles. Each cycle is further divided into 2 to 3 distinct steps of temperature. Most PCRs are standard with cycles having 3 temperature steps. First initiating step is a single temperature step at a temperature greater than 90 degrees Celsius called as the hold, which is followed by cycling, and cycling is further followed by another hold step at the end for the final product elongation or short storage. The temperatures and the span of the time given in each cycle are variable and is dependent upon several parameters such as enzyme used, concentration of divalent ions and the quantity of dNTPs in the reaction and the GC content, which in turn influences the melting temperature of the primers. Let us now talk about the initialization step. This initial step is required for DNA polymerase, which become active by heat application upon them. For the type of polymerase that does not rely on the heat activation, this step is not included. This step is called hot start PCR and involves heating the mixture at a temperature in the range of 94 to 96 degrees Celsius, which requires 1 to 10 minutes. The second step, that is the denaturation step. It is the prime repeated cycling step that compromises the heating of the mixture containing the template DNA at 94 to 98 degrees Celsius for 20 to 30 seconds and can extend up to 1 minute. This heating results in disruption of the hydrogen bonds among the purine and pyrimidine complementary bases, also called the melting of the DNA, thereby resulting in single strands of DNA. Since GC pair by the 3 hydrogen bond in comparison to 2 of the AT pair, GC rich sequences have higher melting point. The third step, that is the annealing step. Following the melting step, temperature is now decreased to about 50 to 65 degrees Celsius for 20 to 40 seconds that allow the joining of the primers, of course with the complementary sequences, to the single stranded DNA template. Annealing temperature for any PCR is about 3 to 5 degrees Celsius below the temperature of the primers used. Since primer sequences are alternate to the template strand, hydrogen bonds are formed between the primer and template DNA. The polymerase then binds up to the primer template hybrid and initiates the DNA synthesis. The next step is now the extension or the elongation step. In this step, temperature and duration to be used rely on the kind of the DNA polymerase taken for the PCR and also on the quantity of the amplification required. The optimal activity of Taq polymerase is the temperature of about 75 to 80 degrees Celsius and 72 is the most common temperature that is used. Through the help of enzyme, a new DNA strand opposite to the template DNA strand is formed by placing the dNTPs in 5 to 3 direction adjoining the 5-phosphate group of the dNTPs by the 3-hydroxyl group of the liberated dNTPs at the terminal of the elongated DNA strand. Under optimal conditions with no dearth of the reactants, 1000 base per minute will be polymerized by the DNA polymerase. With each duplication of this step, the DNA targets amount gets doubled exponentially of the specific DNA fragment. The final elongation. After the last PCR cycle, a temperature in the range of 70 to 74 degrees Celsius for 5 to 15 minutes is provided to make sure that no single stranded DNA is prevented from getting fully extended. The final hold. This step at 4 to 15 degrees Celsius can be done for undefined time and may be employed for the sake of short span shortage of the reaction. A diagrammatic representation of one cycle of PCR which has been explained. Following each cycle, the amount of DNA is doubled under ideal conditions. The components and their own regulatory temperatures of a characteristic PCR cycle are also explained. PCR is generally followed by agarose gel electrophoresis for determining the integrity of the PCR products. The length of the PCR product DNA fragment called amplifier or amplicon is determined by assessment from the electrophoresis band of PCR product DNA against a DNA ladder, which is a set of DNA sequences with known molecular weight or size. The two are simultaneously electrophoresed on the agarose gel alongside the PCR products. Despite the simplicity of the underlying process and improvements in technical, in practice, PCR can fail for multiple reasons. A PCR must be calibrated to work under specific conditions. Most of the time, this is due to occurrence of contamination from irrelevant and extraneous DNA. Such troubles can be addressed with modifying lab protocols and procedures. This is seldom an issue in molecular biology laboratories with a dedicated space for PCR setup and for analysis or purification of PCR products. Use of disposable plasticwares and thorough cleaning the work surface between the reaction setups further reduces the chances of contamination. A very important issue confines to primer based techniques that are usually key factor in enhancing PCR product yield, and spurious product is also avoided. Use of polymerase enzymes with specific buffer components that are suitable for the given enzyme help in the amplification of long or problematic regions of DNA. Nonetheless, PCR is a relatively simpler technique and has high sensitivity and specificity. Amplification of DNA by the PCR. As both the strands are replicated during PCR, an exponential increase in the number of copy of the DNA is observed. Suppose if just single copy of DNA is present before the PCR starts, then after running one cycle there will be 2 copies of DNA. Again after the next cycle, that is the second cycle, there will be total of 4 copies of DNA and so on. Most PCRs are carried to about 25 to 35 cycles. So, considering a PCR reaction with 30 cycles will theoretically have 2 to the power 30 copies of the target DNA. The various stages of the PCR. This PCR process can be classified into the 3 substages. First, the exponential amplification. Under the optimal conditions, reactants, that is the primers and the dNTPs, are available in high concentration, resulting in doubling of the quantity of the product. The reaction at this stage is very responsive and little quantity of the DNA is required. The initial phases of PCR are regarded as exponential amplification and the size of the replicated DNA doubled per cycle. The next, the leveling off stage. Over the time with repeated cycles, there exists some loss in the activity of the enzymes as the use of the reagents like dNTPs and primers make them to become restrictive, and thus the reaction pace slows down. The next, the plateau. The reactants are exhausted and no more products are synthesized due to unavailability of the reagents and or the loss of enzymatic activity. During a traditional clinical results review, the responsibility of laboratory reviewer is to determine whether the baseline as well as the threshold for a given PCR is appropriate. Observations of anomalies in the response are significant in expression of making a conclusion. This is of particular relevance in a specific modification of PCR called Quantitative PCR or real-time PCR and can be used to interpret the test conducted for diagnosis for a disease and omitting the false results. Modifications of the basic PCR technique. Many modifications of the basic PCR technique have been done and are aimed at specific outcomes, yet all these modifications are based on the basic principles discussed in the previous section. The allele specific PCR. This investigative or cloning technique relies on single nucleotide polymorphism, also called as SNPs, which are different at single base in sequence DNA. Knowledge of sequence of a DNA is required in this process that includes the differences in alleles and uses primers whose 3' end includes the SNPs. Under the stringent conditions, PCR amplification is worthless if a difference among the template and the primer occurs. Therefore, successful PCR with an SNP specific primer signals can be done only if specific SNP is there in the template sequence. Assembly PCR or the polymerase cycling assembly. This is rather not as much frequently called PCR modification, where from a pool of long oligonucleotides along with short overlapping segments, synthesis of DNA, which is long, is achieved. The oligonucleotides are opposite in among sense and anti-sense directions, and the order of the PCR fragments is analyzed by the overlapping segments, thus producing the specific long DNA product. Asymmetric PCR. In asymmetric PCR, preferentially either from the two strands of the DNA double helix amplifies. This technique is commonly practiced for those cases where a requirement of multiplication of only one out of the two complementary strands is present. Overload of the primer is required in this technique to conduct amplification of merely one of the two alternate strands. Later, additional cycles of PCR are essential as slow amplification occurs after the limiting supply of primer as they have been consumed. Helicase dependent amplification. It requires a steady temperature instead of cyclic variations in denaturation and annealing extension cycles. The unwinding of DNA by thermal denaturation is replaced by the use of DNA helicase, which is an enzyme that unwinds DNA. The hot start PCR. It helps in lessening of the non-specific amplification in the initial stages of the PCR. It is manually performed by heating the reaction mixture to do the melting temperature, example 94 degrees Celsius, and this reaction mixture should be devoid of polymerase, or if a reaction mixture contains the polymerase, its activity is inhibited at ambient temperature by developing specialized enzyme systems. It is done either by attaching an antibody or by inhibitors use. New fused polymerases, which are dormant at ambient temperature and are right away activated at elongation temperature, can also be used to achieve hot start cold finish PCR. Inter-sequence specific PCR, also called as ISSR. This is commonly practiced in forensic laboratories as a method of PCR for DNA multiplications of the regions among simple sequence repeats to generate a unique fingerprint consisting of amplified fragment lengths. Inverse PCR. The flanking sequences near the genomic inserts are commonly identified by this method. It includes a chain of DNA digestions and self-litigation that results in known sequences at any one terminal of the unidentified sequence. Methylation specific PCR, referred to as MSP. This method is used to detect methylation of CpG islands in the genomic DNA. Treat the source DNA first by sodium bisulfate that converts unmethylated cytosine bases to uracil, which is recognized as thiamine by the PCR primers. Then carry out 2 PCRs on the alternate DNA using identical primer set except at any CpG islands among the primer sequences. One primer set identifies DNA with cytosines to amplify methylated DNA, while the other one identifies the DNA with uracil or thiamine to amplify unmethylated DNA at these points. Mini primer PCR. It uses a heat resistance polymerase enzyme that extends short primers, or we can say small oligos, that is 9 to 10 nucleosides. This allows small primer binding regions to be targeted by the PCR and also to amplify preserved DNA sequences. Multiplex PCR. In this type of PCR, many primers sets inside a singular PCR mixture are required to produce different size amplicon which are definite to different DNA sequences. At the same time, multiple genes can be targeted by this. A single test run, which otherwise would have required several times the reagents as well as further time to perform. Optimization of annealing temperatures is required for each of the primer sets so as to work correctly within a single reaction. Nested PCR. By minimizing background caused by indefinite amplification of DNA, this enhances the specificity of DNA amplification. In 2 continuous PCRs, 2 primers are used. DNA products are generated in the first reaction using one pair of primer, which may still comprise of non-specific amplified DNA fragments in addition to the intended target. The product obtained in the first reaction is used in the next PCR with a different set of primers. Target DNA molecule sequences comparatively in large amount is required in nested PCR. Quantitative PCR, referred to as the Q-PCR. This is the most significant PCR methodology used for quantitative analysis of a PCR product, commonly in real time. Initial amounts of DNA, cDNA or RNA have been quantitatively measured by it. Q-PCR is generally used to analyze the presence of a particular sequence of DNA in a sample along with its copy number in the sample. In addition, Q-PCR is extremely precise and requires the use of fluorescent dyes. Reverse transcription PCR, abbreviated as RT-PCR. Most PCR reactions are conducted with the DNA templates. Reverse transcriptase, that is an enzyme, is used for the amplification of DNA from RNA. It reversely transcribes the RNA into cDNA, which is further amplified by PCR. RT-PCR is broadly used in expressing profiling, in analyzing the gene expression or identifying the series of an RNA transcript. Thermal asymmetric interlaced PCR, referred to as the tail PCR. In this, an unknown sequence adjoining a known sequence is isolated. Pair of primers with different annealing temperatures inside the known sequence is used in tail PCR. Amplification in the other direction from the unknown sequence is done by using degenerate primer. touchdown PCR, also referred to as the step down PCR. This type of PCR reduces the non-specific background by steadily decreasing the annealing temperature with progressing PCR cycles. In the initial cycles, the annealing temperature is usually a few degrees 3 to 5 degrees Celsius higher than the Tm of the primers, while it is few degrees 3 to 5 degrees lower than the primer Tm in the later cycle. Large specificity for primer bonding is provided by higher temperatures, whereas more efficient amplification from the specific products that are formed during the initial cycles achieved by providing lower temperatures. Universal fast walking. This is used for genetic fingerprinting and genome walking using a specially designed two-sided PCR instead of that conventional one-sided approach. This uses two primers, that is, one general primer and one gene specific primer. Students, let us now study the various applications of PCR. First, the selective DNA isolation. PCR is used to separate DNA fragments from genomic DNA or RNA by selectively amplifying specific regions. This in turn boosts many methods, example DNA cloning and generation of hybridization probes for southern or northern hybridization by identifying a specific gene or a transcript. High quantity of pure DNA can be derived by the use of PCR, allowing analysis of DNA samples even from very small amounts of the starting material. Second, DNA sequencing. PCR is used for DNA sequencing in order to determine unknown PCR amplified sequences where one of the amplification primers can be used in Sanger sequencing. PCR is also used for isolation of a DNA sequence to execute recombinant DNA. Third, genetic fingerprinting. For identifying a person or an organism, PCR is used for genetic fingerprinting by comparing experimental DNAs through different PCR based methods in forensic science labs. In addition, some PCR fingerprinting methods have high distinguish power, can be used to identify the genetic link up between individuals such as the parent child or between siblings are also used in paternity testing. The same technique can also be used in determining evolutionary relationships among organisms having similar phylogeny. PCR can amplify the selective regions of target DNA only, thus extremely small amounts of sample can be analyzed, which often is difficult for forensic analysis if only a trace quantity of DNA is available as an evidence, for example, the hair. Analysis of age or how old the given DNA samples is also done by the use of PCR. Quantitative PCR methods are used to estimate the amount of specific sequences present in a sample, that is a technique applied for determining gene expressions level quantitatively. Real-time PCR is a well-established technique for DNA quantification in which after each round of PCR amplification, the quantity of DNA product accumulated is measured. PCR aids in early diagnosis, leukemia and lymphomas and many other malignant diseases, and thus is currently highly used in cancer research and has been routinely used for diagnosis and screening of these cancers. Non-cultivable, that is slow growing microorganisms such as the mycobacteria or the anaerobic bacteria present in animal models and tissue culture assays can also be identified by PCR. PCR diagnostic applications include in microbiology for discriminating non-pathogenic from the pathogenic strains with the help of specific genes and detection of infectious agents. Similarly, PCR can detect the viral DNA as well. A specific set of primers are used at the targeted sequences in the DNA of a virus. Detection of virus is done after and before the infection, as PCR has a high degree of sensitivity. Quantification of the virus can also be done by PCR based DNA quantization techniques. PCR is an advanced and precise molecular biology technique used for amplifying DNA in vitro. Carey Mullius invented this technique, and his contribution was acknowledged by the Nobel Prize after 10 years of its invention. PCR requires a thermally stable DNA polymerase enzyme, divalent ions, buffer and primers which are complementary to a template DNA sequence. A PCR reaction consists of multiple cycles. Each cycle is composed of several steps, namely the denaturation, annealing and extension or polymerization step. Initial stages of PCR are marked with an exponential rise due to abundance of reactants and the follow up in the linear phase where DNA duplicates with every cycle. With the exhaustion of reactants and the accumulation of DNA, PCR's efficiency reduces, and this stage is called the plateau phase. PCR is typically carried out in an apparatus called thermal cycler, which is based upon the Peltier effect, and the temperature is quickly regulated in it by providing a rapid heating and cooling of the tube slots for the amplification of the DNA. Multiple applications of PCR are used widely in the field of molecular biology, clinical diagnosis, forensic sciences and many more. The easy use and high throughput output has made PCR an indispensable technique for almost all the laboratories.